Okay, so welcome to this next video in which we are discussing the use of the phosphodiesterase inhibitors to treat asthma. Okay, so we are in the process of trying to understand why inhibiting the phosphodiesterase 4 enzyme within bronchial smooth muscle cells is going to produce relaxation of those bronchial smooth muscle cells. Okay, so we've seen that the inhibition of the phosphodiesterase 4 enzyme by, for instance, theophylline, which is a non-selective phosphodiesterase inhibitor, or rolipram, which is a selective phosphodiesterase 4 inhibitor, is going to result in an increase in cyclic AMP within the cytoplasm of the bronchial smooth muscle cell. Now, this is going to lead to the activation of protein kinase A enzymes, and these protein kinase A enzymes are going to phosphorylate certain protein targets. Okay, so let me show you the structure of one of the targets that they are going to phosphorylate. Okay, so one of the targets that they're going to phosphorylate is something known as the BK channel. Okay, and this stands for the big conductance potassium channel. So B is for big conductance, and the K is for potassium channel. Okay, so this is the BK channel, which is the big conductance potassium channel. Now, oops, nearly wrote Chanel. Okay, channel, and the big conductance potassium channel is an example of what's known as a calcium-gated potassium channel. Okay, so calcium-gated potassium channels are often abbreviated to CGKCs for short. C for calcium, G for gated, K for potassium, since that's the periodic table symbol, and C for channel. Okay, so let's now discuss the structure of these CGKCs. Okay, so let's say this is the let's say this is the phospholipid bilayer. So these calcium-gated potassium channels are potassium channels. So let's draw a nice channel structure here. So they're permeable to potassium and they're activated to open by calcium. Okay, so here is our calcium-gated potassium channel. Now the calcium-gated potassium channel is not just one protein. It is 4 proteins stuck together. In fact, it's more than 4 proteins stuck together. But initially, we'll just draw it as 4 proteins stuck together. So the main portion, well, the pore-forming unit consists of 4 proteins stuck together. And if you just have these 4 proteins stuck together, that will make a functional calcium-gated potassium channel. You can stick extra auxiliary subunits on, which can modify the function of it, and I'll discuss those in a moment. But this is the core structure that you need. Okay, so these 4 subunits that I have stuck together here are known as the alpha subunits of the calcium-gated potassium channel. Okay, now, basically, there are many different alpha subunits. You might think it would be extremely simple if there was just one alpha subunit, the calcium-gated potassium channel alpha subunit, and all you had to do was make 4 copies of this protein and then stick them together, and that would make you the pore-forming unit of a calcium-gated potassium channel. But it's not that simple. Instead, there are many genes that can all code for an alpha subunit of the calcium-gated potassium channel. Okay, so these are grouped into 5 families, okay? So overall, there are going to be 8 genes for this alpha subunit, and they're grouped into 5 families. So let's summarize these 5 families. Where should I put them? I'll put them here. So there is the KCA1 family, which stands for calcium-gated potassium channel and the first family. There is then the KCA2 family, then the KCA3 family, okay, then the KCA4 family, and finally the KCA5 family. Okay, so these are the 5 families. Now, let's talk about the genes within these families. So in the first family, the first gene is known as the KCA1.1 gene. Okay, and there's only one gene within that family. So this is a gene, this KCA1.1 gene is a gene which will can be transcribed and then translated, transcribed into some mRNA. The mRNA will then be translated, and it will make a protein which can be used as an alpha subunit in a calcium-gated potassium channel. Okay? In the second family, it's slightly more complicated. There are 3 genes within the second family. So, there is the KCA 2.1 gene, there is the KCA 2.2 gene, and then finally there is the KCA 2.3 gene. Okay, so all 3 of these genes all code for alpha subunits that can be used as a quarter of a calcium-gated potassium channel. Then let's move our attention to the KCA3 family. In the KCA3 family there is only one example basically. There is the KCA 3.1 gene. Okay? And then KCA4 family, there's then 2 examples in here, which are the KCA 4.1 and the KCA 4.2. And then finally, the final one is in the KCA5 family, and this is just called the KCA 5.1 gene. So these are our 8 genes which all code for alpha subunits of calcium-gated potassium channels. Now, the question then turns to how can you actually construct calcium-gated potassium channels from this? Do you take one gene, use that 4 times to make 4 identical alpha subunits, stick those together, and that's your calcium-gated potassium channel? Or can you mix and match? Can you, for instance, take some of this gene, some of this gene, and some of this gene, and make mixed alpha subunits? So maybe make an alpha subunit of this gene, an alpha subunit of this gene, an alpha subunit of this gene, maybe another alpha subunit of this gene, stick them all together and make that a calcium-gated potassium channel. Well, you certainly can't do it quite as extremely as I just said. Okay, so within families you can form heteropentmers, and we'll discuss this in a moment. Okay, but between families you can't. Okay, so let's start with KCA1, the KCA1 family, because this is the star of the show. This is how we're going to produce big conductance potassium channels. Okay, so we will use this gene KCA 1.1, and we have no choice basically. We have to create what is known as a homotetramer. Okay, because we can't mix it with any other of the genes because they're in different families. You can't mix across different families. I cannot use the KCA 1.1 gene to make 2 of these alpha subunits and then use the KCA 2.1 gene to make the other 2 alpha subunits. You can't do that. If you're in the KCA2 family, you can mix and match. So you could have 2 alpha subunits made of the KCA 2.1 and then the other 2 made of the KCA 2.2. Okay, you can mix and match between members in the same family, but you can't do that between families. Okay, so KCA 1.1 is the only member of the KCA1 family, so it doesn't have any other alpha subunits that it can mix and match with, so it's forced to make homotetramers. So you'll put 1 KCA 1.1 gene protein in here, 1 KCA 1.1 protein here, 1 KCA 1.1 protein here, and 1 KCA 1.1 protein here to create what is known as a KCA 1.1 homotetramer. And this structure is what is known as the big conductance potassium channel. Okay, so when you stick 4 KCA 1.1 alpha subunits together, that makes you the big conductance calcium-gated potassium channel. Okay, now just for a complete discussion of the calcium-gated potassium channels, let's just discuss a few more of these. So, for the KCA2 family, this is another very famous family of calcium-gated potassium channels. Okay, you can either make homotetramers, which is where you take 4 alpha subunits which are all identical and are all coded for by just one of these genes. Okay, so you either make 4 KCA 2.1s, 4 kCa 2.2s or 4 kCa 2.3s, and you stick all 4 of them together to make homotetramers. So there are 3 possible homotetramers that you can make, but you can also mix and match within the family to make heterotetramers, okay? And whenever you make either a homotetramer or a heterotetramer out of the proteins in the KCA2 family, then the channel that you get is what is known as a SK channel, okay? And this stands for small conductance potassium channel, okay? So this is a small conductance potassium channel. So there isn't just one small conductance potassium channel, okay? There are many small conductance potassium channels because, for instance, the KCa 2.1 homotetramer would be considered a small conductance potassium channel. The KCa 2.2 homotetramer will be considered a small conductance potassium channel. The KCa 2.3 homotetramer will be considered a small conductance potassium channel, and all of the heterotetramers would also be considered a small conductance potassium channel. So when you mix and match, for instance, take 2 KCa 2.1s and then bind them to 2 KCa 2.2s, that would be a heterotetramer, and that would still be considered a small conductance potassium channel, or an SK channel. Okay, the final little family that I want to discuss is this KCa3 family. This is another very famous one. This, again, there's only one member of the family, so you don't have any choices here. All you can make is the KCa 3.1 homotetramer, where you make 4 copies. Whoops, that's an odd M. Where you make 4 copies of the KCa 3.1 protein. Okay, bind them together to make the KCa 3.1 homotetramer, okay? And this KCa 3.1 homotetramer is what's known as the IK channel, okay? And this is the intermediate conductance potassium channel. So this stands for intermediate conductance, so that's the I stands for intermediate conductance, and then the K stands for potassium, and then on the end we've got channel. Okay, so these 3 types of channels, BK channels, SK channels, and IK potassium channels, you'll hear a lot about them. They're quite famous. Okay, so this is how they're actually composed of these calcium-gated potassium channel subunits. Okay, right. Before we go any further, let me just tell you something about the difference between the BK calcium-gated potassium channels and the SK and IK potassium channels. Okay, so the IK channels and the SK channels, these will open in response to elevations in cytoplasmic calcium. So if calcium goes up in the cytoplasm, this activates the SK and IK channels to open, and they'll then allow potassium to move through them. Okay, so they are calcium sensitive, but they cannot be activated by voltage. Okay, so changes in the electrical potential difference across the membrane, they're not bothered about that. They're not going to be induced to open by changes in the electrical potential difference across the membrane. So they're voltage insensitive. This is the difference between the SK and the IK, the SK, IK, and the BK channel. So these two are calcium sensitive and voltage insensitive, whereas the BK channel is both calcium sensitive, okay, so it can be activated to open by calcium, but it can also be activated by voltage changes, so it can be activated by depolarization of the electrical potential difference across the membrane. Okay, so that's the difference between the SK, the IK, and the BK channel. Okay, obviously there's also differences in the conductance, which is reflected within the name, okay? So big conductance potassium channels, when they're open, the amount of potassium that will actually move through them in a certain amount of time is going to be much bigger, so their ability to conduct potassium is much higher, whereas small conductance potassium channels, when they're open, and the amount of potassium they're actually going to conduct is very small, basically, and IK channels are somewhere in the middle. Okay, right, so let's now turn our attention to the BK channel completely. Okay, so we've seen that a BK channel is a homotetramer of 4 KCa 1.1 alpha subunits. However, they are slightly more complicated than that. In all calcium-gated potassium channels, you can also add in beta subunits. Okay, so let me show this. Here are the 4 alpha subunits bound together in this homotetramer here. Okay, but you can also add in beta subunits onto that. So you pick 4 alpha subunits, and then what you can do is stick on beta subunits here. And the beta subunits sort of stick in between 2 alpha subunits, the junction between 2 alpha subunits. So there'll be another one down here. Okay, and you're going to stick 4 of these on. Okay, so here's another one, and there's another one over here, like so, and then another one back there. So these are the beta subunits, and there are 3 beta subunits: the beta 1, beta 2, and beta 3, and they affect the properties of the channel. Okay, so you can either put 4 beta 1 subunits there, or 4 beta 2 subunits there, or 4 beta 3 subunits there. So these beta subunits assembling onto the structure of the calcium-gated potassium channel can affect its properties, basically. Okay, so let's now look at the structure of these beta subunits, and also let's have a look at the structure of the KCa 1.1 protein. Okay, so remember, we have stuck together 4 alpha subunits, which are all KCa 1.1 proteins, and we've also now attached these beta subunits on. So let's have a look at what the actual transmembrane structure of a KCa 1.1 protein is. So they have a structure that looks like this. Here's the amino terminus here. Okay, then they have a single membrane spanning alpha helix, then another membrane spanning alpha helix, the 4th one, the 5th one, the 6th one, then they have a little P loop, and then the 7th one here. So they span the membrane 7 times, and they have this loop that tries to span but fails. Okay, then on this side what they have are 2 important domains, which I'll draw here, like so, before you finally have the carboxylic acid terminus over here. So these 2 domains here are known as the RCK1 domain and the RCK2 domain. Okay, so the RCK in the RCK domain name stands for the regulator of conductance of potassium domain. Okay, so you have the regulator of conductance of potassium domain 1, and then you also have the regulator of conductance of potassium domain 2. Okay, so the R is for regulator, the C is for conductance, and then the K is for potassium here. Okay, so these are the regulator of conductance of potassium domains. Okay, and they assemble into a little ring that's on the cytoplasmic side, basically. So on the cytoplasmic side of the receptor, the RCK domains assemble into a ring, basically. Just like there's a ring of the alpha subunits like this, you have a ring of RCK domains. So let me take a cross-section across here and show you the structure of this. So what you will have is a pore down the middle through which the potassium will move. And then this basically is an octamer of RCK domains. So each alpha subunit is going to put both of its RCK domains into this structure, and each alpha subunit is going to provide therefore 2 RCK domains, its RCK1 and its RCK2 domain, and there's 4 alpha subunits, so you're overall going to have 8 RCK domains. So let me make this crystal clear by colouring in the RCK1 domain in orange, and then the RCK2 domain in blue. Okay, so for instance, if we start off with this subunit back here, okay, this will have both an RCK1 domain and an RCK2 domain. Here is its RCK1 domain in this octamer of the RCK domains. Here is its RCK2 domain. Okay, so those are the RCK1 and RCK2 domains of this back alpha subunit over here. Then this next alpha subunit, it will contribute its RCK1 domain and also its RCK2 domain. Okay, so we've now got 4 RCK domains, and this process will continue. So the next one along will contribute its RCK1 domain and then its RCK2 domain, and then the final one right back here will also contribute its RCK1 domain and then its RCK2 domain. And that's how you get this octamer of RCK domains, which forms a ring at the intracellular aspect of the BK channel. Okay, now these RCK domains, specifically the RCK2 domain, has structures within it known as calcium balls. Okay, and these calcium balls are structures where calcium can come and bind. Okay, so if calcium goes up in the cytoplasm, calcium can bind to the calcium balls on the RCK domains, and this will then trigger the opening of this BK channel. Now, just to complete our discussion of the BK channel before we discuss what protein kinase A is actually going to do to this channel, let's discuss the structure of the beta subunits. So the beta subunits have a very simple structure. They have 2 membrane-spanning alpha helices. Here is their amino terminus, and here is their carboxylic acid terminus there. So 4 of these will assemble around there, and they'll provide this loop that's on the extracellular aspect. This will sort of fold over onto at the top of the channel structure there. Okay, right. So now, what is protein kinase A going to do? Well, protein kinase A, the catalytic subunits of protein kinase A, which are now free in the cytoplasm, free of their regulatory subunits, they're going to phosphorylate the intracellular aspect of this BK channel, which, remember, consists of 4 KCA 1.1 alpha subunits and 4 beta subunits, which could be any of the 3 there. Okay, and then what is that going to do? Well, it's going to increase the sensitivity of this channel to both calcium and also to voltage. So basically it means that the amount of time that these BK channels are going to spend in the open state when phosphorylated is overall going to go up. So you increase the sensitivity to calcium and you also increase the sensitivity to voltage. So the likelihood that this channel will be open is going to increase, basically. Sensitivity to calcium goes up, and also the sensitivity to voltage is also going to go up. Okay, so you're going to have a huge number of these BK channels in your cell membrane of your bronchial smooth muscle cell. Okay, now all of them have had an increase in their sensitivity to calcium and also their sensitivity to voltage, which means that they have a greater chance of being open. So the number that will be open at any one time is going to be greater now that they've been phosphorylated, and therefore the amount of potassium that these things will be conducting is going to be greater. Now, which direction are they going to conduct potassium? Well, in the cytoplasm you have a concentration of potassium that is 155 millimolar or there around, and outside the cell you have a concentration of potassium which is around 4 millimolar. Okay, so the concentration gradient is going to favour the movement of potassium out, but we have to consider another factor, which is the electrical potential difference across the membrane. Okay, so let me just remind you of what electrical potential difference actually means. So, firstly, let's discuss what electrical potential is. So electrical potential is this concept in physics. Basically, it is a mathematical structure that helps us to understand reality. Okay, so the idea is this, that you ascribe to every point in three-dimensional space a real number, and this can be positive or negative, and this is called the electrical potential. So every point in space has an electrical potential. Now, the electrical potential of all the points that are outside the cell will roughly be the same, and the electrical potential of all the points inside the cell will roughly be the same. However, the electrical potential inside the cell, i.e. of all the points inside the cell, is not going to be the same as the electrical potential of all the points outside the cell. So we can measure the electrical potential outside the cell, which I will call E for electrical potential, and then I'll put O for outside. And we can also measure the electrical potential inside the cell. Okay? Right, and these two numbers are not the same. We can then construct an electrical potential difference, okay, also called a voltage, and you therefore have to give a direction. You have to say the voltage from somewhere to somewhere. Okay, so when people talk about the electrical potential difference across a membrane, they always mean the electrical potential difference from the outside of the cell into the inside of the cell. So they mean directed in this direction. Okay, so what they mean is if a little man was standing in the outside of the cell measuring electrical potential, he would measure a certain number, this number here. Okay, if he moved to the inside of the cell, he would measure a different number, which is this number here. He can then ask, how much has the number on my screen changed when I moved from outside to inside? This is what is meant by the electrical potential difference across the membrane. Okay, people drop the from outside to inside because they assume you understand that. Okay, so this means the electrical potential inside, the new electrical potential, minus the old electrical potential. Okay, so this is what is meant by electrical potential difference across the membrane. It means the electrical potential inside minus the electrical potential outside. But really you should think of it as, if I moved from outside to inside, how much would that number on my screen change, where this screen is measuring electrical potential? Okay, right. So the electrical potential difference across the membrane of a smooth muscle cell is generally around negative 60 millivolts. Now, what does that mean? That means that the insides of the cell has an electrical potential 60 millivolts lower than the electrical potential outside. It means that if you move from outside to inside, your electrical potential, the number on your screen, will go down, and it will go down by 60 millivolts. Okay, so the electrical potential inside is lower than the electrical potential outside. Now, potassium is a positively charged ion. It wants to be where electrical potential is lower, and at the moment the electrical potential is lower inside, okay? It's lower by 60 millivolts. Okay, so the electrical gradient across this cell is favouring the movement of potassium into the cell. So, this I always think is a little bit like Game of Thrones. When the queen says one thing and the hand of the king says another, who wins? Who wins this battle? Concentration gradient says move potassium out. The electrical gradient says move potassium back in. Well, just like Cersei wins, Cersei is the stronger one. The movement out is stronger, and the movement in is going to lose, basically. So the electrical gradient is not strong enough to beat the concentration gradient. Okay, so this electrical gradient tries its best, and it does lower the movement of potassium out. So what you'll have to do is say, this would have been what the movement out if there was no electrical potential difference across this membrane. We're going to now have to subtract the movement in due to the electrical potential difference. So the net movement will be some little arrow like this, which is this arrow take away this arrow. Okay, now if you wanted to stop all movement together, okay, if you wanted the electrical potential gradient to be as strong as the concentration gradient, you would have to take it to around negative 85 millivolts. So it's not strong enough, basically. So you still get a net movement of potassium out. So when you open these BK channels, you're going to get a net movement of potassium out. So now that we've increased the sensitivity of these BK channels and they're open more of the time, you're going to get more potassium moving out of the cell. Now what does this mean? Well, potassium carries a positive charge, so we've now got a movement of positive charge out of the cell that we did not have before. okay? And when you move positive charge out of the cell, that means that you're going to reduce the electrical potential inside, because remember, positive charge raises the electrical potential. Where positive charge is, the electrical potential will be higher. We're now moving positive charge out of the intracellular compartment and into the extracellular compartment. So we're putting in positive charge here, which is going to raise the electrical potential outside, and we're taking away positive charge from the intracellular compartment, which means that the electrical potential of the intracellular compartment is going to go down. So this number is going up, this number is going down, okay? And we were asking what is the electrical potential difference from outside to inside. So what effect is opening these BK channels going to have on the electrical potential difference? Well, if the electrical potential outside has gone up and the electrical potential inside has gone down, well, the electrical potential difference from outside to inside was already negative 60 millivolts, which means that the intracellular compartment was 60 millivolts lower than the extracellular compartment. Okay, but now we're raising the electrical potential of the extracellular compartment and lowering the electrical potential of the intracellular compartment. So the amount by which the intracellular compartment's electrical potential is lower than the extracellular compartment's electrical potential is going to go up. So this number is going to become more negative, basically. Okay, so it's going to become more negative. Okay, and that change is known as hyperpolarization. Okay, now why is hyperpolarizing the membrane going to favor the relaxation of the bronchial smooth muscle cell? Because if you think about this for a while, you'll come to an uneasy conclusion. Why should it affect the contraction of the bronchial smooth muscle cell? After all, the contraction of the bronchial smooth muscle cell was induced by histamine and cysteine leukotriene. So let's just go through this. So if we have our bronchial smooth muscle cell here, okay, we have these H1 receptors on the surface of our membrane, and we also have these cysteine leukotriene receptors on our membrane. So this is a cysteine leukotriene receptor, either type 1 or it's a cysteine leukotriene receptor of type 2. This is an H1 receptor. And these, remember, resulted in the activation of the GQ pathway, okay, which overall leads to the production of IP3, inositol 1,4,5-trisphosphate, which then led to the release of calcium from the intracellular stores. No part of this seems to require anything to do with the electrical potential difference across the membrane. Okay, so why is hyperpolarizing the cells stopping the release of calcium from the intracellular stores? Well, basically, this pathway is oversimplified. Okay, so let me explain. So, everything has to do with the IP3 receptors. Okay, so let's have a look at the structure of an IP3 receptor. Okay, so an IP3 receptor is a massive great receptor in the membrane of the endoplasmic reticulum. So let's say this is the endoplasmic reticulum, or the ER for short. Okay, so it's a massive great receptor that can allow calcium to move through it from the endoplasmic reticulum lumen here into the cytoplasm. Okay, so it can allow the release of the intracellular stores of calcium, and therefore this calcium that's released can then drive the contraction of the smooth muscle cell. Okay, right, so the IP3 receptor itself is again a tetramer. It's made of 4 separate proteins glued together. Okay, now there are 3 separate genes which can code for a subunit of the IP3 receptor. Okay, so there is the IP3 receptor gene 1 all the way up to the IP3 receptor gene 3. Okay, and you can either make homotetramers or you can make heterotetramers. So you can either make a IP3 receptor where you stick 4 IP3 receptor 1 proteins together to make an IP3 receptor 1 homotetramer, or you can make heterotetramers where you mix and match. Okay, another fun fact about these IP3 receptors is that they are absolutely massive. Each one of the subunits of the IP3 receptor has around 2,750 amino acids. Now take it from me, that is a massive, massive protein, and you are sticking 4 of these things together. These are massive proteins. They're not as big as the ryanodine receptors, but they are still big. Okay, right, now let's talk about how they open, because this is where the answer to this problem lies. Okay, so let me draw the top of an IP3 receptor. So this is the cytoplasmic aspect of an IP3 receptor, or at least it's my cartoon representation of a cytoplasmic aspect of an IP3 receptor. Okay, so the current model for this is that you have 4 calcium inhibitory binding sites initially available on the surface of the IP3 receptor. Okay, so every single one of the subunits of the IP3 receptor has an inhibitory calcium binding site. Okay, so now if calcium binds in this inhibitory calcium binding site, it will make the probability that the channel will open even lower, i.e. it will inhibit the IP3 receptor. Okay, now all of these IP3 receptors also have an IP3 binding site, which is in blue here. So now what can happen is when you produce IP3 by the GQ pathway, okay, IP3 will come and bind to this IP3 binding site. So 4 IP3 molecules are going to need to come in. 1 will bind to the IP3 binding site of each of the 4 subunits. So 1 IP3 molecule will bind here, 1 here, 1 here, and 1 here. What that then triggers is a change in the structure of these IP3 receptor subunits, and this is the really crucial portion to understand. You do not, repeat, you do not open the IP3 receptor. Okay, so when IP3 binds, it does not cause the opening of the IP3 receptor. That's the important thing to understand. That's where this model is oversimplified. Okay, previously you might just have believed that IP3 bound to the IP3 receptor and triggered it to open, and to some extent it does, but well, it doesn't, but there is some. It's a good oversimplification, basically. It's a useful oversimplification. If you don't want to have to explain the full thing, that's a useful thing to just assume, basically. Because usually when IP3 binds, it does then, you know, what follows is the IP3 receptors open and you release calcium. However, IP3 binding to the IP3 receptor does not cause it to open. However, you'll see why it's a useful oversimplification. Okay, so what will change is you will remove that inhibitory calcium binding site. Okay, so when the IP3 receptor subunits change conformation, the inhibitory calcium binding site will be removed and it will no longer be exposed to the cytoplasm, and it will be replaced by a stimulatory calcium binding site. Now, if calcium comes and binds to the stimulatory calcium binding sites, so again you'll need 4 calcium ions because you've got 4 stimulatory calcium binding sites. Okay, if 4 calcium ions come and bind to the stimulatory calcium binding sites, then the IP3 receptor will open and it will release calcium from the endoplasmic reticulum into the cytoplasm. Okay, so IP3 does not open the IP3 receptor. Instead, it primes it so that calcium can bind to the IP3 receptor and cause the IP3 receptor to open. So before IP3 binds, you can't open the IP3 receptor. But once it binds, you're then in a position that if calcium also comes along, it can now activate the IP3 receptor to open. Okay, so this is the important part now. You don't just need IP3 to open the IP3 receptor, you also need calcium. Now, usually, where does this calcium come from? Well, there is some calcium in the cytoplasm of cells, but it's really, really low, and the chance that you're going to get 4 calcium ions concurrently binding to all 4 of these stimulatory calcium binding sites with this tinily low calcium concentration is very, very slim. The concentration of calcium in the cytoplasm is 100 nanomolar, okay? That's low. Compare that to the extracellular concentration of calcium, which is 1.5 millimolar. Okay, so this is 15,000 times higher than this. So, what is generally believed is that in order to get the calcium that is going to activate these IP3 receptors, some calcium must come in from the extracellular fluid, okay, and this is probably let in by voltage-gated calcium channels. And I should emphasize that this stuff is still being researched. It's not a fully understood topic, but the speculation, and the most likely correct speculation, the speculation that most people would back, is that what has to happen is voltage-gated calcium channels also have to open. They have to allow calcium to move in from the extracellular fluid, and this calcium that comes in from the extracellular fluid will then bind to the primed IP3 receptors, the IP3 receptors which have got IP3 bound and therefore have exposed stimulatory calcium binding sites. And then when the calcium comes in from the extracellular fluid, it will bind to the stimulatory calcium binding sites, and then that will activate the IP3 receptor to open and release calcium from the ER. And it's the calcium that's released from the ER that will then trigger the contraction. So you don't let in much calcium from the extracellular fluid. Instead, you let in enough and spray it onto the surface of the primed IP3 receptors, and then that triggers the release of calcium from the ER, and that releases a huge amount of calcium into the cytoplasm, which then triggers the contraction of the smooth muscle cell. Okay, so if these are voltage-gated calcium channels, and the abbreviation for voltage-gated calcium channel is VGCC, then they need depolarization of the electrical potential difference across the membrane to open them. Now, if we've hyperpolarized the membrane by opening these BK channels more and allowing potassium to leave the cell, then the chance that these voltage-gated calcium channels are going to open is going to be slimmer, okay? So, it will damage the ability of the histamine 1 receptor and the cysteine leukotriene receptors to cause release of intracellular calcium, because although you will still produce IP3 and you'll prime the IP3 receptors to open, if there's no calcium, then they're not actually going to open. It doesn't matter that you've got IP3. So it's believed generally that what opening the BK channels does is it hyperpolarizes the cell, and the hyperpolarization inhibits the smooth muscle cells by stopping the entry of calcium from the extracellular fluid into the cytoplasm, which is then going to drive the release of calcium from the ER. So without this calcium, you're not going to get release of calcium from the ER, and without calcium release from the ER, you're not going to get contraction of the smooth muscle cell. Okay, so that's how taking these prophylactic phosphodiesterase 4 inhibitors, whether they're selective, such as rolipram or roflumilast, or non-selective, such as theophylline, that's how this is going to be protective against bronchial smooth muscle cell contraction. And remember, it's also useful because it stabilizes the mast cells and stops them from releasing as much mediator. Okay, so both of these actions are how these oral phosphodiesterase 4 inhibitors are going to protect against asthmatic attacks.